Hey everyone, Professor Davis here, and we are going to start to dive into genetics, and this is going to be over a series of videos. And so this first part, we're going to look at patterns of genetic inheritance, and these patterns are going to mostly follow Mendel's studies. So Gregor Mendel was the kind of scientist who is termed as the father of heredity. He lived between 1822 and 1884, and he was an Austrian monk. He's the one who formulated the laws of genetics in the 1860s, and he was proven right for the most part in all of his studies. His work is still repeatable today. He used just the scientific method, being objective, and he also had a very strong mathematical background that helped him be able to be successful. He was lucky in the plants that he chose to work with. As an Austrian monk, he had particular jobs that he was to focus in to help the monastery, and his was in the garden. And so he worked with gardening, but he picked the true breeding garden peas. Now he lucked out here. He picked them for a reason. They were very easy to grow. He could cross-pollinate them very easily, and they had a very short generation time, so it didn't take too long for the plants to grow before he could then see the results of what he was doing. And so he had his reasons, but his pick of these plants was also very helpful in that they followed very true, simple genetics. Now, he postulated his theory of inheritance, and it replaced a very popular theory at the time called the blending theory. The blending theory said that offspring's traits were inherited from the parents, and it blended together from the two parents. So one of the best examples here is like the red carnation and the white carnation, and when those were mated together, the result were pink flowers, because that's a blend of adding white and red together to make pink. And so this is what they were kind of basing some of their information on. The problem is then when you were to take a pink flower and made it with another pink flower, you would think that they would all be pink because there was nothing to really blend and they'd all be pink flowers, correct? The problem is in this blending theory, this is not what was happening. When you crossed a pink flower with another pink flower, sometimes you got red and white flowers back, which did not prove the blending theory. And so this is what kind of got Mendel thinking about what this means. And he came up with his theory of inheritance. So Mendel's new theory of inheritance was published in 1866, but it was largely ignored at the time. Most biologists at the time were not used to thinking in mathematical terms and using the math that he came up with his structure and all. So even Darwin missed the significance of Mendel's work during this particular time. But by the 1900s, meiosis had been actually observed, and 3 plant geneticists realized that chromosomes in meiosis provided the physical, actual explanation for Mendel's results. So even though his results were shown mathematically and that sort of thing, meiosis actually being visualized and seeing it actually supported all of the stuff that he had come up with without seeing it like these particular geneticists had. So Mendel looked at 7 different traits in his pea plants, allowing him to compare more than one phenotype over many generations. And these were the different traits he was looking at: flower color, flower position, seed color, as well as seed shape, the pod shape, as well as the pod color, and even the stem length. Okay, and this is where he ended up getting a lot of his information. And he was able to determine which traits were what we call dominant and which traits we call recessive. So inheritance of dominant and recessive alleles can explain Mendel's crosses. Dominant alleles may mask or hide the expression of a recessive allele. So this is one thing that he ended up seeing. So if the dominant allele was present, it's going to hide the fact that the recessive allele may still be there. He demonstrated that the inheritance of certain traits in pea plants follows particular patterns, and these patterns are actually termed as his Mendelian inheritance. Okay, and so again, this is where the mathematical side came from with the idea of statistics and bringing up patterns. So let's look at Mendel's kind of laws of heredity that he was able to come up with. And the first law that we want to look at is called the law of segregation. So this is when an allele pair segregates or separates from each other during the process of meiosis. Okay, and so segregation means that they're separating. So an individual has 2 factors for each gene, because remember we have the 2 chromosomes. Okay, you have 2 chromosomes for say number 1, those are going to separate individually. Okay, so this one's going to separate one way and this one's going to separate the other. So this is because individuals are diploid. They're going to have to separate when we do those haploid cells, egg and sperm. So these factors known as genes are going to separate during the formation of the gametes, and chromosomes separate during the process of meiosis, specifically meiosis during anaphase 1, because in anaphase we're going to see that split, that first split into 2 separate cells. And so this is where we're going to see that segregation. Gametes can only contain one factor or one gene for each trait. So as a result of meiosis, gametes are haploid. Okay, so again, this is why meiosis proves what he was saying. He didn't call them genes and stuff at the time. They were traits, but it meant the same thing. So each gamete only receives one allele of each pair because the pairs were separated. Fertilization provides each new offspring with 2 factors again. Remember, as fertilization is we bring a chromosome 1 from mom and a chromosome 1 from dad, and they come back together. They're going to see that you have 2 back together. So fertilization restores the number of chromosomes back to that diploid number. Okay, so again, remembering that we have to, for sexual reproduction, have to have meiosis and fertilization, but segregation had to occur first. There had to be a separation during meiosis first. The next law that he came up with was the law of dominance. So of the 2 factors, the 2 genes that are present, one codes for a particular trait and it will be expressed, known as the dominant allele, whereas the other is the unexpressed or recessive allele. So if the dominant allele is present, you're gonna see it. This is what he's telling us. So if the dominant allele is present, you're going to see it. The dominant gene, though, can mask the expression of the recessive. Now, this doesn't mean you'll never see the recessive. It just means that if a dominant is there, it's going to hide it. But a recessive trait can only be expressed and seen if the dominant gene is absent. So if the dominant gene is not present and we have 2 of the recessive, you will see that recessive trait. Okay? But if the dominant is there, you're going to see the dominant trait. This is what the law of dominance says. The next trait we want to look at is the law of independent assortment. Now, this means that each pair of alleles is going to assort or segregate when it lines up. They're going to assort and get ready to segregate independently, okay, of the other alleles. Chromosome 1 does not care what chromosome 5 is doing. They don't care how they're lining up or anything like that. Chromosome 1's only worried about itself. It's independent. Chromosome 3 doesn't care what chromosome 12 is doing. Okay, they're going to line up independently and they are going to then separate independently. So during metaphase 1 of meiosis, where they all line up in the middle at the equator, the homologous pairs are going to align randomly. So it might be chromosome 1 and then now we have chromosome 15 and then you've got chromosome 10 and then you have chromosome, say 7. They're going to all line up independently. They don't care what the other chromosomes are doing. Okay, so the example of this is the gene for eye color is independent of height. It's not saying that, oh, all blue-eyed people are going to be short and all brown-eyed people are going to be tall. No, they're independent of each other. However, some genes on the same chromosome may be found together, and those are known as linked genes. We have found that red hair and freckles kind of go together. But guys, this is because they're found on the same chromosome. One's not found on chromosome 2 and the other one found on chromosome 5. They're both found on chromosome 2. In that particular case. Now, I don't know if that's actually the chromosome they're on, but I'm giving an example. Okay. So if they're found on the same chromosome, this is different. But if it's different chromosomes, the different chromosomes don't care what the other chromosomes are doing. So all possible combination of a factor or gene can occur in a gamete. So gametes are not genetically identical and they contain a variety of gene combinations. And this is what's great because meiosis leads to genetic variability, or what we consider variation. All right. And so this is a great thing that gives us a lot of diversity. So we have the law of segregation, we have the law of dominance, and we have the law of independent assortment. So what is inheritance? Inheritance, guys, is the process by which characteristics of an individual are passed to their offspring. All right. And so taking those individuals genetically from me and giving them to my children. Okay. So it's inheritance. So there is a study of this inheritance process. We can see it's a study of, again, heredity, relatedness, variation in genes, individuals, populations, and species, and those would all be under that umbrella of genetics. So let's kind of take a look at Mendel and how he looked at the parent generation. So Mendel's P means parent generation, his F1 generation, which is the offspring off of the first generation, and then the second generation. So say we're looking at two traits or alleles. In this case, we're going to look at tall and short. Okay. So the capital T is going to represent tall and the little t is going to represent short. Now this is important because the capital letter is going to represent the dominant trait. So in this particular case, tall is dominant over short. So in the parent generation, the original parent line, they took two purebred or true breeding plants. In this particular case, he took a true breeding tall plant, which was big T, and a true breeding short plant, which was little t, little t. And the only way you can see that it's short is if it has the two little t's. Okay, so we have these two true breeding plants, and he was going to mate them. What he found is that the offspring produced from the parent generation were hybrids. Now, a hybrid means you're combining two different things, and so this means that all of them were big T, little t. So he found that they were hybrids, big T, little t. The issue, though, here is that they were all tall, because the big T told you that the tall was present, which was masking the short. Okay, so now what he did is he took two of the plants from that F1 generation and he mated them. So a hybrid crossed with another hybrid. When this happened, he found that three came out as dominant or tall and one came out short. Okay, and so this told him that the recessive trait was still present. So this tells us the recessive trait was still present. It was just being hidden in that F1 generation. It just didn't go away completely. Is going to be made out of a double helix of DNA, which we talked about back in the chemical chapter. This double helix of DNA is going to create proteins. So when we look at this, the whole idea of the nucleotides are going to help create those genes, and then genes are going to help create those alleles. Okay, and so this is the hierarchy. We're going from the chromosome, which is the big picture, down to the alleles, which is the details. Okay, so that's what we're getting into, is those details. So guys, when we look at the gene is a physical location on a chromosome that codes for a specific product. A gene here is known as the unit of heredity. Okay, so it's the unit of heredity. The locus is a gene's physical location. It's its location on the chromosome. So the locus is the gene's location. The allele is the one member of a pair of specific genes. So an alternative form of the gene could be present, but this is the allele. Okay, so there's normally at least two genetic variations when we look at alleles. In this particular case, you're seeing that one is yellow and one is red. But those are the alleles. They code for the same gene because they're found in the same location or locus. Okay, and so this is what we're looking at when we break down the idea of that chromosome. So it takes two genes to express a trait. That's why you had to get one from mom and one from dad. It takes two genes to express the trait. Multiple genes are found on each chromosome. Okay, so chromosomes are going to have lots of genes all on the same chromosome. So in man, there are approximately 25,000 to 50,000 genes. These are all found on the 23 pairs of chromosomes. Each chromosomal pair carries specific traits, and that's where the Human Genome Project came out and mapped out those traits. What's crazy about this Human Genome Project is that they've mapped them out, but they still do not agree on the actual number of genes that are present. Okay, they don't really agree on this, and further research is still being done. So alleles are going to be described as either being dominant or recessive. So a dominant gene is the one that's expressed if it's present. So this gene, if it's there, you're gonna see it. And so we represent this with the capital letter when we're talking about the genes. So it's gonna mask the expression of the recessive, and it gets denoted by a capital letter, like an example, the big T, capital letter T. On the other hand, recessive genes, they are only gonna be expressed if the dominant gene is absent or not present. This is where the gene is masked by the dominant allele. This is denoted by the lowercase or small letter, the little t, lowercase t. Okay, so dominant alleles are represented in capital letters. Recessive alleles are represented in lowercase letters. Now, some terms to describe this. If the offspring have two identical alleles, they are what we call homozygous. So homo means same, and in this case they have the same alleles. Now this could be that they are big T, big T. They have two dominant, so these would be considered homozygous dominant organisms, or they have two little t's and they would be considered homozygous recessive. Okay, these are also the ones who are known as being purebred because they are exactly the same. They have the exact same letters, two big T's or two little t's, so they're considered purebred. On the other hand, heterozygous are offsprings with two different alleles. Hetero means different. And so in this case, they have a capital letter and a lowercase letter. So they're big T, little t. They're different. This is also known as a hybrid, as they're a combination of two different things, a big T and a little t. Now, based on our dominant and recessive, since the big T is present, the dominant trait is what's being seen. But the recessive trait here is being hidden or masked in a hybrid. Okay, so homozygous are the same. Heterozygous, they are different. Another term we need to look at are genotype versus phenotype. Genotype refers to the genetic makeup of an offspring. So we could say they're homozygous dominant, or they're homozygous recessive, or they're heterozygous. Okay, it's going to be looking at those letters. So homozygous dominant, they're big T, big T. Homozygous recessive, they're little t, little t. Heterozygous, they're big T, little t. Now, do we describe people like this? Or animals or things? No, we don't. We don't look at that plant and we say, oh, that plant's little t, and big P, little p, and whatever. No, we don't. Okay, we describe individuals or organisms with what we call the phenotype. Guys, the phenotype refers to the physical appearance of the offspring. What do they look like? So instead of saying that plant was little t, and big P, little p, we would say, oh, that is a short plant with purple flowers. Okay, and so we are going to give the description of their appearance here. So if big T is tall and little t is short, big T, and big T, little t would be tall because the dominant trait is present. But little t would be short. And so this is how we describe individuals, is by their phenotype. However, guys, their phenotype is determined by their genotype. So your phenotype is determined by your genetics or your genotype. So a diploid organism's two alleles may be the same or different. So if they are homozygous, they are going to have the same pair. So this would be blue and blue or brown and brown. We're talking about eye color here. Okay. Heterozygous would be different pairs alleles, and so in this case it would be blue and brown. Okay, so if we look over here at individual A, B, and C, which individual would have the blue and blue where they are homozygous? Well, blue and blue would be C. What about brown and brown? Well, that's individual B. So which one is going to be heterozygous, meaning different, blue and brown? That one would be A. Okay, and so we can describe those individuals. Now, the genotype is the combination of alleles carried by an organism, so full heredity code of the allele, whether expressed or not. But remember, the phenotype is the observed trait. So we'd say, oh, they have brown eyes or they have blue eyes. Okay. So we're going to use the alleles as either big B or little b. Now, if you'll notice, big B in this case is brown in color. This means that brown is dominant over blue. Blue is recessive. So what is the genotype for individual A? If individual A is heterozygous, they are going to have one brown and one blue, so they are big B, little b. Okay? But what about person B, individual B, that's homozygous dominant? Because they have brown. This would be big B, big B. And then individual C is homozygous recessive, and so they are going to be little b, little b. Okay, so you guys see how the genotype can be determined? Now let's talk about the phenotype. What color eyes do the individuals have? All right, so let's skip over to B first. B is both dominant, brown and brown, and so B is going to have brown eyes. So now let's go back over to individual A. Since brown is present there, but blue as well, brown is dominant, so they're going to still have brown eyes, but they are a carrier of blue. You just don't see it. On the other hand, if we want to see the blue eyes, they have to have the two little letters. And so C is going to have blue eyes. So you guys see how we can utilize the information to not only find the genotype, but also the phenotype. Now this brings us to something that Mendel did, which were crosses, where he crossed two individuals and he predicted to see what was going to happen. So the first type of cross we're going to talk about is a monohybrid cross. Mono means one, so we're crossing one trait here. So monohybrid cross, we're crossing one trait. Now remember, it takes two genes to express a trait. So genetic problems are a way for us to statistically predict what the offspring's genotype and phenotype may be. So this involves probabilities, which talks about math. Okay. Now I'm going to teach you a really easy way to look at these, but we're going to look at monohybrid crosses. Now we're going to briefly look at these, but in my class, you're also going to end up doing some genetics problems, and I will have a separate video that goes over how to complete those genetics problems. But this is still going to give you an overview and help you start the process. So when we look at these monohybrid crosses, we have to use what we call the Punnett square when doing genetic problems with offsprings. So when we look at Punnett squares, we have to create this square-like structure where we have 4 boxes that are present. Now, with these boxes, we're going to put the alleles that were created in the different egg and sperm from the parents on the top of these alleles. And so this is where those gametes are going to come into play. So we place those letters based on what could have been available based on those genetics. Each of the boxes then represent the genotypes of the offspring, because what we do is we bring the information down from the top and over from the side for each of the boxes. This allows us then to be able to determine with 2N what the genotype of the offspring is, and then in turn what the phenotype will probably be. Okay, and so this is what it looks like with the whole idea of egg and sperm, and we're using the Punnett square. So the law of segregation, and we're going to do a Mendel example here. He had true breeding plants. Okay, and so he had a true breeding green pod and a true breeding yellow pod. So green is dominant to yellow, and so if we were looking at this, this would be like big G crossed with a little g. What he found is that in the first generation, they were always the dominant trait. In this case, all of them were green. So where did the yellow go? Okay, well, it didn't disappear, because when he crossed the two F1 generations, he saw that there was going to be some green and a yellow that still came up. This is going to show that a pattern was found. Okay, so find a pattern here, and we see that there is going to be a pattern that does occur. And again, the Punnett square is useful in this. So dominant and recessive monohybrid cross using the Punnett square, we're going to look at the trait color for pod color. So green is going to be dominant, and we're going to represent that by a big G. Yellow is recessive with the little g. 2 individuals that are homozygous for each allele are crossed. So I have a big G crossed with a little g. So let's figure this part out. Okay, so this is how we would create it. I take those 2 big G's and I separate them, and I put them each into a column. So big G, the first big G is in the first column, second big G is in the second column. I am then going to take those 2 little g's and I'm going to separate them on the rows. First little g is on the first row, second little g is on the second row. Okay, so now I have my gametes. So now what do I have to put in the box? When I go through this process, I'm going to bring the big G down and I'm going to bring the little g over. In this particular case, all the boxes are going to be exactly the same. And guys, you always list the capital letter first. So every box here was big G, little g, which means since big G was present and it's the dominant trait, all of the offspring are green. Okay, so this is how he found it out using the Punnett square. So all individuals of the cross have the same genotype, therefore they also have the same phenotype. Okay, so now we are going to take 2 of these individuals and we're going to cross a big G, little g, with a big G, little g. Okay, so here's the F2 generation. So you're going to notice that the gametes look a little different. I'm going to take that first big G, little g, and I'm going to separate them. The big G is going to go in the first column, Little g is going to go in the second column. I'm going to then take the second big G, little g, and I'm going to separate them on the rows. I got big G in the first row, little g in the second row. So now what goes into the box? So if I'm going to bring the big G down, I'm going to bring the big G over. The first box is going to be big G, big G, big G. If I go to the second box on the first row, I'm going to bring big G over and bring little g down. I have big G, little g. If I go to the second row, first column, I have big G, little g. And my last box is going to be the two little g's. So I'm bringing down and over, down and over. That's what we're doing when we are doing these Punnett squares. So in this case, we now have three different genotypes. Okay, I have big G, which is homozygous dominant. I have big G, which is heterozygous. And little g, which is homozygous recessive. So we have the three genotypes. However, there's still only two phenotypes. If the big G is present, they are green in color. So I have three that are green, and I have one that has only recessive, which means that the color would be yellow. Guys, this creates a ratio. 3 boxes to 1 box. We call this a 3-to-1 ratio of the second generation. 3 boxes to 1 box. Or like you could say 75% to 25%. Okay, because if I have only 4 boxes, I've got 4 quarters. Okay, so this is a ratio. So here's an application. Mendel's law of segregation. If the parent genotype is big T, little t, then the possible gametes are going to be a big T or a little t, because during meiosis, they separate. So let's set up this Punnett square with our t's. I've got a big T, little t crossed with another big T, little t. So how am I going to set this up? Well, first of all, I'm going to take the first individual and I'm going to separate their gametes. I'm going to place big T on the column 1, little t on column 2. I'm going to take the second individual and I'm going to separate their alleles into their gametes for the rows. Big T on the first row, little t on the second row. Okay, doing the Punnett square, I can fill in the boxes. Okay, so the first box is going to be big T, big T. Second box is going to be big T, little t. The first box in the second row is going to be big T, little t. And the second box in the second row is going to be little t, little t. All right, so I got a couple of questions here. What is the chance of getting either a big T or a little t? Well, it's half, right? So it's a 50% chance. All right, because if I have a big T and a little t both being present, I only have 2 alleles, there's a 50% chance I'm going to get a big T, and there's a 50% chance I'm going to get a little t. So what is the chance of producing a recessive offspring? So recessive are the 2 little letters. What are the chances based on our boxes here that we're going to get a recessive offspring? Well, it's only 1 box out of how many? 4. So it's 1 out of 4. And in this case, if I put that into a percentage, that is 25%. 1 out of 4. So the next one, what is the chance that you would have a recessive offspring or a homozygous dominant offspring? So it's asking me to look at 2 different things. Well, this is known as the sum rule of probability. If it's asking, can you have this or this, I've got to add them together. So I already know that I have 1/4 of a chance of getting a recessive, 1 out of 4. What's the chance of me getting a homozygous dominant? Well, let's break that down. Homozygous means same. Dominant means capital letters. This is talking about the big T, big T. So what are the chances I get a big T, big T? That's also 1 out of 4. Well, what's 1/4 plus 1/4? Well, that's 2/4. 2/4 can then be reduced to 1/2. And so that's a half a chance. Well, in percentage, that's 50%. Okay. The next one is, what's the chance of producing 2 recessive offspring? Now, if I want 2 of the same thing, this means this and that. The and makes it different. The and means I have to use the product rule of probability, which means I have to do multiplication. The chance that I am going to get a recessive offspring is 1 out of 4. Every time it's 1 out of 4. So now I got to do 1/4 times 1/4. When I do 1/4 times 1/4, what's 1 times 1? 1. And what's 4 times 4? 16. There is a 1 out of 16 chance that both of the offspring that the plant has are going to be short or recessive in this case. So you have the rule of addition. If it says this or this, I'm going to add the fractions together. If it says this and that, I need to do multiplication. I'm going to get the product of. Okay, so this is just giving you an idea of how you can go about with the mathematics side of it. Okay. All right, so let's look at the idea of the law of probability. Probabilities of the phenotypes and genotypes of offspring may be expressed in different ways. We can do a ratio, dominant, recessive. So in the case of our last example, we had 3 dominant to 1 recessive, so you could do 3:1. You could also do a fraction. It's the number of the specific trait over the total. So like this is what we did with that, where we had 1 out of 4. Okay, so that's a fraction. You could also divide the numerator by the denominator in the fraction and multiply by 100 for the percentage. So 1/4 is going to be 25%. So these are the different ways you could potentially represent what we're seeing with the Punnett square. So guys, probabilities don't change with each offspring. Just because I had a short the first time doesn't mean I get to take that off the table. The probabilities don't change with each offspring because, guys, heredity has no memory. Your body's not sitting there and being like, oh, they had a short kid last time, I need to give them a tall one this time. Nope, they don't remember that stuff. Okay, so it's going to repeat every time in the sense of having the same probability, the same chance. Probabilities, guys, are only predictions. It's not reality. However, with the more and more offspring you have, the closer and closer you get to the actual statistics or probability. So the more times you do it, the more chances you're going to get to that, like boys versus girls, 50-50. Okay, and so this is what we're looking at there. Okay, it gets closer and closer to that prediction, that probability. So guys, this brings us to one test cross, and the purpose here is used to determine the genotype of an individual expressing a dominant phenotype. Remember, if something has a dominant phenotype, like in this case it's tall, it could be big T, or it could be big T, little t. So how can we know what the genotype is? Well, we're going to match it up with a recessive trait because we know that recessive's genotype. Okay, so this is the case here. I have a big T, but I don't know what the second letter is. I don't know because it's dominant, so I can't tell. I'm going to cross that with a recessive, little t, and I'm going to see what happens. Okay, so I'm going to go over here to the second one. I'm going to take that big T. I'm going to pair it up there with those little t's. Our first 2 boxes we should know, and in this case, they would both be tall. Okay. It's what happens in the second set of boxes that's important here. Okay, because we know that little t's are coming into the picture, but what's the next letter? Okay, if they are little t's as well, this tells us that the parent was big T, little t. So if you have an offspring with recessive phenotypes, if you end up getting some short plants out of that cross, this means that the parent that we weren't sure about is heterozygous. It's big T, little t. Because you have an offspring that is going to show the recessive phenotype, and that can only happen if both parents give a recessive. Okay, and you see that here in the picture. On the other hand, over here, if I do the same thing, my first 2 boxes are exactly the same as what we saw over there. Okay, for my first column. It's my second column that I have to take a look at. Okay, so I still put my little t's, but if all the offspring come out tall, all the offspring come out with the dominant trait, this tells you that that particular organism was homozygous dominant. They were a big T, big T. So you guys see how the Punnett square could also help us work backwards a little bit? Knowing what the offspring are, we could figure out what the parents are. Okay, this is what we do with a test cross. Now I'm going to introduce one other type of cross to you. This is known as a dihybrid cross. Now, di tells you that we are going to be looking at 2 traits. In this case, guys, we're tracking 2 different traits located on 2 different locations. These 2 traits are also considered non-linked, meaning they're not found on the same chromosome. They're on different chromosomes. So guys, this means they're not linked. If I'm looking at this, let's say we're looking at flies. I'm going to look at their wings versus their body. These are 2 traits I'm going to look at, but they're not linked together. They're on different chromosomes. They're on different locations. Okay, so they're not linked. If they were linked, they would be found on the same chromosome. Okay, now they're not found in the exact location on that chromosome, but they are found on the same chromosome. We're going to take a look at the ones not linked. Okay, so let's just say that this particular fly is big L, little l when it comes to their wings, and big G, little g when it comes to their body. This means when we separate these, green doesn't care what yellow is doing, but when they separate, one of each trait is going into that egg, or one of each trait is going into that sperm. Okay, this is that idea of independent assortment, and then they're separating, which gives you segregation. So since we're looking at 2 traits, I can't just do 2 separate Punnett squares. I've got to do Punnett squares that take both traits into consideration. Now, a normal Punnett square has 4 squares. If I'm looking at 2 traits, I've got to do 4 times 4, which gives me how many squares? Ooh, this Punnett square is going to have 16 squares. It's going to be a big Punnett square. So let's take a look. I'm not going to ask you to do very many of these, but I want to introduce them to you so that you can see. So what would happen if he studied a plant that differed in 2 traits? Okay. This is going to be a dihybrid cross. So he's going to look at 2 traits. He's going to first look at the pod color, and he's going to look at the seed color. Now, this is weird because pod color dominant is green and pod color recessive is yellow. But seeds are opposite. Seed color yellow is dominant, whereas green colored seed is recessive. So I'm going to take 2 pure breeding plants and I'm going to mate them, and then that's going to give us our hybrid. Okay, so it's going to give us our hybrid. So green pod color and yellow seed color are cross-pollinated with a yellow pod color and green seed. Green pod color big G and yellow seed color big Y are dominant. Yellow pod color little g and green seed color little y are recessive. This results in offspring that were all heterozygous for green pod color and yellow seed color. So they're heterozygous because they have a big G little g and a big Y little y, but they are still green pods with yellow seeds. So here's my experiment. I've got a dominant and recessive dihybrid cross using a Punnett square. I'm going to look at pod color and seed color. So green pod color is big G and yellow seed color is big Y. They are dominant. Yellow pod color is little g and green seed color is little y. They're recessive. We're going to take 2 individuals that are heterozygous for each alleles. So I've got a big G, big Y, little y, crossed with big G, big Y, little y. All right, so here is how I would set up the Punnett square. Okay. So I'm going to end up doing a thing that we call kind of like foiling in math. I'm going to take that first capital G and I'm going to pair it with each Y that I can. Not with the little g because the little g has to separate from the big G. So I'm going to take this big G and I'm going to put it with the big Y. And then I'm going to take the big G and I'm going to put it with the little y. Has it now paired with every Y that it can? Well, yeah, because there was only 2. I'm now going to take that little g and do the same thing. So little g is going to be paired with big Y. And then little g is going to be paired with little y. And that gives me the 4 across the top. That was the first parent. The second parent, we're going to do the same thing and we're going to put them down the side. So big G gets paired with big Y, and then big G gets paired with little y. Little g gets paired with big Y, and little g gets paired with little y. This is going to then help us put in the proper genotypes into each box. So in this first one, I'm going to bring the two big G's together and the two big Y's together. So this one is big G, big Y, big Y. So what color are they? Well, green pod, yellow seed, because they have all the dominant traits. Okay. But in my next box in that row, my next box in that column is going to be big G, big Y, little y. You guys see where those two big G's came together and the big Y came down, the little y came over. This one is still green and yellow, but it does have one recessive Y. The next one in the column is going to be big G, big Y, little y. Again, still green, still yellow. The last one, big G, big Y, little y, still green, still yellow. So this whole first column still looks all the same, even though genetically they look a little different. So let's go to the second row. The second row, I'm going to have my big G, big Y, little y. So I've got green and yellow. But guys, look at the second box in that second column. I have two big G's, which means the pod is still green, but you guys see how there's two little Y's there? This means now the seed is green. You also see in the third box, you have big G, big Y, little y, so it's still green and yellow. But in the last box, you see big G, little y, still green, but green seeds. So you see how we're starting to see some differences? In the next row, we have big G, big Y, still green and yellow. We have big G, big Y, little y, green and yellow. But this next one, we have two little G's, which makes the pod yellow, but two big Y's, which makes the seed still green, which makes the seed still yellow. We also then have big G with big Y, little y. So I have a yellow pod and a yellow seed. In the last column, you will see that we have big G, big Y, little y. That is green and still yellow. But then you have big G, little y. So I have green pod, green seed. Little g, big Y, little y, yellow pod, yellow seed. And then last but not least, all the recessive little g, little y, little y. This guy is yellow with a green seed. If you counted up the boxes that had green pods and yellow seeds, there are 9 boxes with green pods, yellow seeds. There are 3 boxes who have green pods and green seeds. And you have 3 boxes that have yellow pods and yellow seeds. And then your last little box down there in the corner has yellow pod and green seeds. Guys, this is a 9:3:3:1 ratio. In other words, we can say it's a 9:3:3:1. So we still can detect a pattern that's present here. So this is a consistent ratio of a 9:3:3:1 in the F2 generation of this particular dihybrid cross. So guys, the big thing here is that this brings into effect all the laws that Mendel talked about. We have the laws of segregation, we have the laws of independent assortment, and we also have the laws of dominance, all shown here in this particular Punnett square. So let's do one more example of a dihybrid cross. In this case, I'm going to use wing length and body color in flies. Okay, so wing length, dominant is long, whereas recessive is short. So I'm going to use a big L for long, a little l for short. In body color, gray is dominant over black. So big G is going to be gray, little g is going to be black. So what's the genotype of a long-winged, black-bodied fly? Well, guys, the long wing means there has to be one big L, but it could be another big L or a little l. It doesn't matter. That's why I put the question mark there. It just depends. But to be black-bodied, they have to have 2 little g's. What about the genotype of a short wing gray body? Well, in this case, they have two little L's, so they have short wings. But the gray body means that they have a capital G, but we don't necessarily know what the other G has to be. What about a long-winged gray body? Well, as long as they have a dominant L and a dominant G, it doesn't matter what the other two are. But the last one, guys, is the recessive. If they're short-winged with black bodies, they have to have two little L's and two little G's. That is the only option that they have. The only option that they have. So let's do our law of independent assortment here. Okay. Here are our parents. I have a big L, little l, big G, little g. So this is a long-winged, gray-bodied fly. And I'm crossing it with a short-winged, black-bodied fly because there's two little L's and two little G's. Okay. So I need to make the gametes for my first one. So big L, first big L is going to go with the first big G. And then big L is going to go with the little g. Now big L has been paired up with the two it can. So little L is going to then go with the big G. And little L is going to then go with the little g. That's the first parent. The possible gametes for the second parent are really easy since they're all lowercase. I have a little L with a little g on all of them. So now I'm going to set them up on my Punnett square. Okay. So I have my big G, sorry, my big L, my big G, my big L, little g, little L, big G, little L, little g. Down the side, I have my little L, little g's in each of the rows. Okay. Now we're going to start pairing up the boxes. So in the first box, I have big L, big G, little g. Okay, so I always put the L's together. I always put the G's together. Capital letter always goes first. Okay. Now, guys, what about the next box in this column? It's going to be exactly the same. Since all of the gametes on the side are the same, what you put in the first box is going to be down that whole column. So in this particular case, guys, all four of these boxes are going to have long-winged, gray-bodied flies. Hey, what about in the next one? The next one, I have big L, and two little g's. If this is the case, all of these four in this column are going to be long-winged, but they have black bodies. The two little g's make them black-bodied. The next column is going to have two little L's, a big G, and a little g. Now, the two little L's tell you that they have short wings. The big G tells you they have gray bodies. And last but not least, you have all the recessive. Two little L's, two little G's. In this case, you have short-winged, black bodies. So guys, if you'll notice here, every column is different. Every column has four boxes. This creates a 4 to 4 to 4 to 4 ratio. In other words, you can reduce that to a 1 to 1 to 1 to 1 ratio. I got 4 long and gray, 4 long and black, 4 short and gray, and 4 short and black. Okay, and so this is how you end up doing those Punnett squares. All right, so last one on here is how would you determine what the genotype is of a long-winged fruit fly with a gray body? Now, we did a test cross earlier. We know that it has to have at least one big L, right? Because that's long-winged. And we know that it needs to have at least one big G for it to be green-bodied, but we don't know what the second letter is. Well, we have to mate it with something we do know, which is little l, big little g, little g. So little l, little g. And we have to look for recessive offspring. If recessive offspring end up being present, they are recessive for that other trait. So I'm going to set up the Punnett square here. So I have big L, big G, because those are the ones I know. I have big L, question mark. I have question mark with big G. And then I have 2 question marks because I don't know what they are. I'm then going to also take that little l, little g, and put it down the side. So depending on what I get, so in this first case, it doesn't matter. I'm going to get big L, big G, little g. That one we already know. I've got to look at the rest of them. If in this next one, I get recessive body color, then that means that this is long-winged and black-bodied. So that would be big G, little g for that particular trait. In the next column, I would also look for recessive. If there's two little L's, that would tell me that they have the big L, little l, which then means it would be heterozygous for the wing length. They carry the recessive. And I definitely would look at this last column to see if they have recessive traits. Guys, if no recessive traits are present, this means that they would be all homozygous dominant. Big L, big G, big G. But guys, this is how a test cross could also be used for a dihybrid cross. Okay, so it still could be used with a dihybrid cross. All right, so I know this kind of gave you a little bit of a crash course into genetics, but I wanted to give you an introduction. Like I said, I will have another video that goes into a little more detail where I show you how to set up those Punnett squares a little bit better. But if you do have any questions or concerns, please let me know.